Administrative Epidemiology Unit. So the leader of the group is Professor Nick Timpson, who's also the PI of AUSPAC, the longitudinal cohort study that's already been mentioned. And we're really interested in understanding the implications of increased adiposity or higher BMI on downstream health, and trying to fill in the gap in the middle. So as I'll kind of go on to a bit more, we kind of know that, you know, high adiposity, or if you want to measure it as body mass index, BMI, is associated with a whole range of diseases, but we don't really know the paths that go in between. So that's kind of the area we're focused on. There's quite a few people in the group doing quite diverse projects. So there's, being in the IU, we've got a lot of focus on genetic epidemiology, but we also have some guys doing wet lab work, in vitro models, and so on. And we're really focused on a kind of triangulation approach, so bringing together information from lots of different study designs, all with, you know, their own limitations and biases, with a view to kind of answer this question about, yeah, what are the downstream health impacts of BMI for our health? Okay, so I don't think anybody's in the room really is unaware of the impact of obesity kind of in the UK, worldwide, in terms of individual implications, but also kind of societal implications. But I just wanted to raise a couple of things that are relevant to the work that we do, that have been kind of, we've seen movement in the last few months. One of them is around the definition of obesity. There's been this paper published recently talking about how we should be defining obesity, and a suggestion of kind of separating the weight effect from the kind of health consequences. So the suggestion that actually maybe we should be talking about clinical obesity and subclinical obesity, depending on whether the health of the individual is affected. And also with a view to thinking about not just about how much weight a person is carrying, but where the distribution is. So thinking about abdominal adiposity versus just general adiposity. And then also, you know, how we're talking about obesity and the language we're using. So I think historically there's been this issue that, you know, maybe personal blame, these people are just, you know, morally inferior, people who just, you know, it's their own fault. We're very much moving away from that, which is brilliant. And that means that we can start to really focus on the biology and what is underpinning this disease and what are the complications and kind of downstream impacts. So yeah, like I said, what we're interested in is finding out why and how BMI is associated with increased morbidity and mortality. So what we know is that, you know, having excess adiposity, however you measure it, we know that there's limitations to body mass index. We can use waist circumference, we can use waist-hip ratio, but that's associated with a whole host, probably hundreds of diseases. But we really don't know what the pathways are that are connecting these things. So some of it, when you look observationally, some of it will be confounding, there'll be shared factors, but there is increasing evidence that at least some of these associations have causal underpinning. In some cases, we have a partial understanding of what goes on. So, for example, reduced insulin sensitivity involved in the path to diabetes, a kind of role for kind of the physical side of things with sleep apnea. So once you get, you know, an increased fat mass around the airways, then that causes that obstruction. But there's other cases where we really have very little information. Psoriasis would be one. Cancer is, you know, a whole host of cancers seem to be affected by BMI, but we don't really know why. And so the research that I'm doing is thinking about, well, can we use molecular intermediates to try and start to unpick these pathways, these biological pathways. So today I'm talking about metabolites. So by metabolites, I mean small molecules. We look at them in the blood, so all the small molecules circulating in the blood. And so that's kind of what we're talking about, is then if you have BMI, if you have variation in BMI combined with these molecular measures, then we can start looking under the hood. So it kind of, you're no longer looking at the outward phenotype, but you're looking at the body's response to that phenotype. And it provides a kind of snapshot in time of the body's kind of metabolism and health. And then hopefully we can identify some biological pathways that are being perturbed by adiposity, by increased weight. Then we can look at the relevance of those biological pathways to some of these diseases. And maybe some pathways will be affecting more than one, others will be pretty unique. And as I mentioned already in our group, we're interested in not just using single study designs, but using multiple different study designs, each with their own limitations, each with their own biases, to try and answer the same question. And that just increases the robustness of our findings. So we've brought together data sets across several different designs, so population studies, studies where we have information about genetic predisposition to increase BMI, and then relevance to my talk today is working together with clinical trials of weight loss interventions as a way of creating that variation in adiposity. So I'm going to talk specifically today about a study we've worked with using calorie restriction to induce weight loss, and then another one looking at surgery. And these are really valuable studies for us because we're generating a change in adiposity within an individual, which helps to account for a lot of the biases we see when we're doing cross-sectional analyses. It's also a pretty radical change in body weight over a relatively short period of time. And you just can't get this kind of in the wild, if you like. It's very difficult to observe in a population level, at least at scale, and without the complication of weight loss caused by ill health, which you don't really want kind of in the mix. So the first one I'm going to talk about is the diet trial. So really fortunate to work with some great clinicians based at University of Glasgow and Newcastle University. So they had this study where they had designed in patients with relatively new onset type 2 diabetes, and they were trialing a weight management program which was underpinned by this Counterweight Plus dietary intervention, which consists of around 12 weeks of a very low calorie diet, followed by 2 to 8 weeks of structured food reintroduction, and then a more long-term kind of help in maintaining a healthy lifestyle and keeping that weight off. And the primary outcomes for the trial were around type 2 diabetes remission and substantial weight loss, so weight loss of 15 kilograms or more. And it was delivered across 49 primary care practices, so GP surgeries across Scotland and Northern England, and each practice was randomized to either deliver the intervention or to deliver the control, which is just kind of standard care for those patients. And 290 patients then fed into the final RCT analysis. So what we were able to do was to take some samples that they'd collected at baseline and at 12 months and send those off for metabolomics analysis. So we had them analysed by 2 different providers, so Metabolon, who operate a mass spec platform for generating metabolomics data, and then Nightingale Health, who use an NMR approach. And these are complementary technologies, so the mass spec gives us semi-quantitative data on over 1,000 metabolites from a single sample, and this covers everything, kind of amino acids, sugars, carbohydrates, but also xenobiotics, so it can pick up drug metabolites and anything really that's circulating, including some things which we just don't know what they are, but we have them measured and we can look at whether they're associated with our exposures of interest. And then the NMR data is fully quantitative, so it's in millimoles per litre. Amino acids, a really good breakdown of lipids, and some of those have actually been clinically validated now, so they could be used in clinic for diagnosis. And like I said before, the key is this weight loss difference. So we are measuring people before and after they lose kind of around 8 kilos in weight. So this is a bit of a side track. When we first got these data, it was the first time I'd worked with metabolomics data, and I just wanted to understand, like, what's the structure of this data, what are the properties, what are the characteristics, before I kind of launch into a statistical analysis. You know, I understand things about my data, and, you know, what QC should I be doing on this? And there really wasn't a kind of gold standard or established way of doing this in the literature. And to be honest, when we looked at papers that had already been published, it wasn't always even clear exactly what had been done. So it wasn't very transparent. That kind of pre-analytical phase between receiving the data and running the statistical analysis was a bit of a mess in the middle. And we knew that we were going to have quite a few data sets coming through from our different studies. So I decided to embark on this idea of having a kind of pipeline, so a pre-analytical pipeline, if you like, to process these data. And what these providers normally give you is an Excel spreadsheet, which immediately fills me with fear, because I imagine people copying and cutting and exporting into CSV and all of these things. So the idea was that the pipeline would extract the data from the Excel sheet and run it through, summarize it, allow you to put some filters on, and all in a way that's reproducible. And at least if something goes awry, you know how that's come about. You don't have that missing bit where people are just manually messing with data. So yeah, a single pipeline. We wanted it to be flexible, so we don't want to always exclude on the same missing this or whatever, because it just depends on the study. And yeah, like I say, at the end, the idea was to have this reproducible and citable And it also means that you don't have to waste a huge amount of your paper writing about, this stuff, because you can just say, okay, here's the code, this is what I did. And so we've now got an R package called MetaboPrep. It's in GitHub. Honestly, it's a bit of a pain now, because as you might imagine now, we had it so that you could take the data they delivered and run it through, and then they changed their deliverables, and now we need to remake the package because it's all broken. So the deliverables used to be a bit tricky to work with, and so we built in lots of things that would help us to deal with that. And then they kind of improved their deliverables, and now all of those little things that we integrated have now started making it break. So anyway, but it's there, and we're working on it. We're working on MetaboPrep v2 at the moment. Okay, so back to the main story. So in the end, so I did quite a lot of different kind of analyses and post-hoc analyses, but I just focused on the main ones. So the main model is pretty simple. I did it in a univariate way, so linear regression, metabolite by metabolite, just to estimate what is the intervention effect. So what's the difference from baseline and the intervention group as compared to the control arm. I also used a logistic regression to look for differences in missingness by trial arm, because some of the metabolites really don't appear in very many people. They tend to be the xenobiotics or the drugs and things like that. And so it's much better to look at actually presence versus absence, because you don't have a really good kind of average level otherwise. And it's really nice to have the control arm, because that allows us to control for anything that might have changed over that period that's not related to the intervention. So, this is quite a short trial, so there's only 1 year difference, but you might expect to see a 1 year aging effect, but that will be cancelled out with our analysis because we have that control group that's just going through standard care. And also any technical things like the time the sample spent in the freezer will be the same in the control arm, so we can account for that. So as you might imagine, the footprint of BMI and BMI change is pretty substantial. So about a quarter of the NMR metabolites were shifted by the intervention, and about 12% of the mass spec. The difference there is partly to do with the fact that the NMR platform is lipid focused, so obviously it's kind of set up to pick up these kind of changes. And then also 19 mass spec derived metabolites were different in terms of their presence at the end. And the strongest signal we saw was for metformin, which was reassuring because that's obviously a drug for diabetes, and these people in the intervention arm, some of those have entered remission, so they're no longer taking their medication. What was interesting was that we saw evidence not only of weight change, but also sustained changes to diet and lifestyle. So it wasn't just about the weight loss, it was about things that people were changed that were carried on up to 12 months. So by 12 months, they're no longer on this kind of strict diet, they're living a kind of a normal lifestyle, if you want, with a normal diet, but you can see changes that they've made compared to baseline. And this figure is just summarizing, so this is all the metabolites that we found associated with the intervention. So each row is a metabolite, each column is an individual, and if you just leave the data to organize itself according to correlations and similarities, then it does tend to cluster those in the treatment group together, so they're in the orange there. But you do have some people who look like they're not in the treatment group, which is what you expect. Some people are just not behaving as everybody else. And then in the black and white columns are indicating the people who achieve type 2 diabetes remission and those that didn't. So again, they tend to cluster together. And you have a group of metabolites that tend to go up in the intervention group and a group of metabolites that tend to go down, shown on there as well. But for me, the tricky thing with the metabolomics data is who cares? Like, what does this mean for my health? We don't really know. We just have this long list of metabolites. We don't really know what half of them do. And so trying to get to that next stage of interpretation is actually kind of hard. The linear model is definitely the easiest bit. So just a few post hoc analyses that I did to try and begin to understand what was going on. So one of the strongest signal actually in the mass spec data was for this biochemical called erythronate. And when I did my Googling, there was very little in the literature about it. It wasn't an obvious candidate. What do I do with this information? And actually the little star means it's a tentative identification. So it's actually even, it's quite a new metabolite. They're not even quite sure what it is. But one thing that we did was to build a kind of hierarchical clustering tree, so to bring metabolites together that behave similarly. And when we did this, when we looked at erythronate, it was then in this kind of family with ribitol and a couple of others, erythritol. So these are common sugar alcohols, and these are present naturally in fruits, but also components of sweeteners. So that kind of made sense to me then, because this looks then to be a kind of diet-based signal. What's interesting is that the super pathways and sub-pathway information is what Metabolon provides to us. That's their kind of part of their annotation of the features. And if you look at those, it wouldn't be immediately obvious that these things were kind of all related, because it's just, you know, the labels that attached are to do with their chemical structure, not necessarily to do with the biology. So that's one thing we did. An obvious question is, you know, how much of this is weight versus other things? Because of the design of this study, on its own, I would say it's pretty impossible to tease out the weight effect, because the weight effect is always correlated with the intervention and all the other elements of the intervention. But just simply by adjusting for weight change in the primary model, we could get some sense of what could be attributed to weight. And in most cases, you see the effect estimate attenuate, but not all effects were related to weight loss, and these are the ones that were clearly, you know, weren't influenced then by weight, and that kind of makes sense. So omega-3 fatty acids are an essential fatty acid. They're to do, you know, the amount it's circulating is to do with how much you've taken in and absorbed, and so it makes sense that that's not downstream of BMI. What was quite nice is around the time that we were about to publish this, actually, there was a publication of a prospective analysis of type 2 diabetes patients. So this was looking at the metabolomic footprint before you get diagnosed with type 2 diabetes. So what's changing in your run-up to that diagnosis? And so in this figure, we've got the relative risk of type 2 diabetes per change in the metabolite on the y-axis, and what the observed effect estimate after intervention on the x-axis. And what you can see is quite nice downward slope. So that means that, you know, you've got things like glucose and mannose up there that are increasing on your way towards a type 2 diagnosis, and then a decrease by the intervention. So you can see that expected relationship there. And then finally, we were interested in, well, what's, you know, when you look at the data, nearly everybody in the intervention arm has lost weight, but only about 50% of them achieve type 2 diabetes remission. So, you know, can we see that from the data? And if we look firstly at glucose, which is kind of the easiest place to start because it's actually involved in the diagnostic criteria for diabetes and remission. And what we can see is, so here on the x-axis, we've got the quartiles of weight loss. So the ones in Q1 have got the greatest weight loss versus the ones at Q4 who are actually slightly weight gain, actually, if anything. And then the difference, the change in metabolite through the intervention. And then in black are those who didn't achieve type 2 diabetes remission by the end, and grey those that did. So what you can see if you just focus on the black is that regardless of how much weight these people lost, they didn't really manage to shift their glucose. Whereas those in the remission arm, those with the grey, you can see that almost kind of linear relationship between the amount of weight that they lost and their decrease in glucose. And we see this for other simple sugars, mannose, fructose, and also this molecule here, 1,5-anhydroglutatol. And it was, when I looked in the literature, this has actually been proposed as a marker for type 2 diabetes as well. So, and the interesting thing for me here is that whilst, you know, weight change is pretty much required in order to achieve remission, it's not deterministic. So there's plenty of people who lose weight and who don't achieve remission, and I'm sure clinicians see this in practice all the time. But for me, this is really interesting because it's when we have variation in response like this, and that's an opportunity to dive in and see what's going on under the hood and kind of biologically. Okay, so that was the diet one. I'm going to talk very briefly about the surgery one. I won't go into detail because I haven't got time, but we essentially did the same thing, but using a surgical trial. So this time, fortunate to work with Professor Jane Bluesky, who led this trial. She's Bristol researcher and surgeon, and the Bristol Trials Unit. So this was a study of, a randomized study of surgical intervention. So they looked at, initially it was set up to look at two different gastric operations, so gastric band and gastric bypass. So people would be randomized to those. But then kind of in the early days of the trial, they realized that gastric sleeve was becoming very popular as a bariatric operation, and so they included that one as well. So it ended up to be three different surgeries. And over the 7 years, they recruited over 1,300 patients, and this was really a really big achievement because it's really hard to get people to be randomized to surgery. There's no control arm in this setting, just the 3 different groups, and you'll notice from the dates it was also impacted by COVID, so it was really a monumental effort on behalf of that team. So basically they would turn up, they would get randomized to surgery, 5 to 6 months later they would have their surgery, and then there's a pretty comprehensive follow-up schedule right out to 36 months. And fortunately for us, they built into a protocol the collection of samples for future research, and that was samples at baseline and then again at 36 months. And again, we've got this massive weight loss over that period of time. And so we're looking at not the immediate post-surgery impact, but downstream when things should have stabilized. So like I say, I'm not going to go through that in detail, but the point of this is that we want to then bring that together alongside our diet data and our diet results and say, okay, well, what's the same here and what's different? And that can help us to begin to unpick. You know, we know from the direct study, we see signals of weight, we see signals of food, we see signals of medication. And to help us kind of work out what's going on, we can then correlate this with, so this on the x-axis is the results from the surgery, and on the y, the results from the weight loss intervention. It's actually flipped, so it's per unit increase in BMI now, because it's easier for us to understand that way around. But basically, you know, where we see things occurring in both, then we have greater confidence that's a weight change. And then when we see things occurring in one, so here we've got beta-cryptozanthin, which is a vitamin A precursor that comes in tropical fruits like oranges, and we only see that signal in the diet intervention. We don't see that signal in the surgery. And what we want to do next is to add in pharmaceutical intervention. So again, probably struggled not to notice the influx of new weight loss medications. And the interesting thing about them is that there's also been, you know, quite a lot in the media and also, you know, in academic work saying, okay, yes, they have a benefit from weight loss, but actually there's evidence that they're benefiting through other pathways as well. So even if you don't get the weight loss effect, you get other health benefits. So this is going to, you know, help us understand that. And finally, just all the other things that I haven't got around to doing that I'd like to do, that if anyone's interested in any elements of this, then give me a shout. So integrating knowledge, so like I said, trying to make sense of some of these associations and, you know, what these metabolites are, what they're doing, so using public databases and the literature. An alternative way to bringing data together, so I showed you today a kind of meta approach where you take results from 2 studies and compare them. But what I was really keen to do was to think about the question of, okay, if you start with obesity, you undergo surgery, you come out, you know, you're lighter, but the question is, are you back on a trajectory towards a healthy weight and a healthy individual, or actually are you now, you've had surgery, you're on some other trajectory, you know? And so what I brought together there in that study was data from Biban sleeve, but also from Mouseback, so they do feature. Because I wanted to see whether these people who had had surgery were moving towards the kind of Ausbach healthy control group, if you like. So, but I haven't really got as far as finishing that analysis yet. I'm keen to do multivariate work. Obviously, you know, none of these changes happen in isolation. I'd rather be talking about a footprint or a network than I would univariate metabolites. So I'm hoping to do some work with Dan Lawson to help with that. And then also the follow-up stuff. So we haven't really touched yet on what the impact of all this is on downstream disease. You know, that's where the kind of genetic and prospective epi can help us. Okay, so thank you all for listening, and just want to acknowledge my great team. So Nick's research group, David. David's a postdoc who's now in the States. Maddie was a PhD student who's just graduated, and Lucy, who's still in the group doing stuff on similar work on proteomics. And all of the teams at the trials. Like I say, it's really, really hard to run these trials. I had quite a lot of kind of first-hand experience with Biban because I was responsible for getting the samples back to site and everything, and it was a monumental effort. So yeah, and any questions, happy to answer. Thank you.